Good evening, everybody. I'm Dr. James King. I'm a senior lecturer in the School of Sport, Exercise and Health Sciences at Loughborough University. I want to welcome everybody to this first session of what is a series of 4 talks that we've put together relating to COVID-19 and lifestyle. Tonight, to kick off, Dr. Martin Lindley, who's an academic within our school, is going to give around an introduction to the immune system and a bit more specific information about COVID-19. And hopefully this more generic talk, which will go on for about 35 to 40 minutes, will set up some of the talks in subsequent weeks quite nicely. And these talks over the next 4 weeks have been developed mainly by academics at Loughborough University, working within the National Centre of Sport and Exercise Medicine. But we do have 2 guest lecturers, one from the University of Coventry and one from Nottingham Trent University, who will deliver talks with us over the next few weeks. I just thought before I introduce Martin that we'd start just by giving a little bit of background to these talks. So last series of 8 talks for managing or treating various conditions that we see commonly within primary care. And these talks are ultimately attended by also lots of different types of allied healthcare professionals. The talk went down really well. People had lots of suggestions for subsequent talks, and we were looking to sort of roll this educational series out further. In fact, we started to put together a bit of a mini conference that we were due to start at Loughborough this year, but then obviously COVID-19 hit, and clearly that wasn't possible, so we put those plans on hold. But we thought, rather than sitting back and doing nothing, we get our heads together, and most of us within the school are following a lot of the research around COVID-19 and certainly lifestyle aspects. And we thought that we could set up a nice series of talks related to areas of our expertise, and that's essentially what we've got. Now, following in from the talks that we had last autumn, we are keeping the same sort of theme of targeting these talks at people with an assumed level of knowledge. So like I said previously, our talks were aimed at general practitioners and allied healthcare professionals. I think it's fair to say that across all of these talks in the next 4 weeks, we're assuming a basic level of knowledge related to kind of the physiology or the metabolism that's involved within the talks. That's not to say that members of the general public won't take anything from these talks. Indeed, we've tried to make sure that even if you have a really basic knowledge, you'll still get something from these talks. But I think it's fair to say that the aim is to assume a basic level of knowledge and really to get some robust science across, but ultimately with clear take-home messages so that the talks are actually usable for individuals. I just wanted to go through a few ground rules. So I think everybody's already got their microphones muted and video switched off. I think we're expecting 250 to 300 people tonight, so obviously if everybody leaves their microphones on or their videos on, we'll have a bit of a problem with bandwidth. Relating to questions, tonight there's only 1 talk, but in subsequent weeks there'll be 2 or more talks. Therefore, in those weeks we'll have questions after each talk, maybe just 1 or 2, and then a larger question and answer session at the end. Tonight, because there's only 1 talk from Dr. Lindley, we'll just hold off obviously to the questions at the end. And Martin said that he's happy to stay around for as long as necessary to answer all the questions that you might have. Just to make you aware as well, these talks are recorded for viewing later. So obviously if you do speak at all by mistake, by switching your audio on, you will get recorded. And I should say actually as well that when you do submit questions, please submit them via the chat function. And when you go onto the chat function, you should be able to select the moderator. So we've got a member of staff who will receive all of your incoming messages and then put them to our speakers, who will then read the questions out and provide an answer. So I hope that's clear. Lastly from me, this is the final slide before Martin comes in. Just to sort of outline to you that, like I've said, there's 4 weeks of talks. As you can see here today is the introduction to the immune system. Next week, I've got colleagues from our school who will talk about exercise and immune function. So Professor Letty Bishop, who's an expert in immunology, will talk about whether exercise can actually help reduce infection risk, particularly relating to viruses. I've got a colleague who's a specialist in nutrition who will also talk about diet and immune function, again with implications for viral infection. And I've also got a colleague who's a cancer biologist who'll talk specifically about immune function exercise in cancer patients. The week after that, on the 7th of July, I'll be back and actually giving 2 talks. So my expertise is around appetite regulation, energy balance. So I'll kick off by giving a talk around COVID-19 and obesity and highlight some of the evidence that we've seen around that. I've got a colleague, Dr. David Clayton, who'll talk about different diets, weight management. Then I will come back in to talk about exercise and weight management, obviously with implications for COVID-19. Then in the final week on the 14th of July, I have a colleague, Professor David Broom from Coventry, who'll talk about physical function in older adults. When these talks were planned, I should say that we were under strict lockdown, and the idea for that talk on physical function was seeing grandparents and individuals who sometimes were quite active all of a sudden weren't able to go out. And there's lots of evidence around reduced activity, reduced functional capacity, which can actually be enduring. And then the final talk there from Dr. Florence Kinnefick, she'll talk about the mental health implications that have come about in light of the COVID-19 pandemic. I believe there's a different link for sessions each week. So if you obviously want to attend any of those sessions in subsequent weeks, please make sure that Alison Stanley, who is our administrator, make sure she's got your email so that she can send out the new links for each session each week. Okay, I won't rabbit on anymore. I'll pass you on to Dr. Martin Lindley, who will go into detail about the immune system. Thanks, Martin.
Thank you, James. I will put my screen up so that we can start screen straight away. And we shall make a start. So, good evening, one and all. As James pointed out, this is an introductory session, really, to give you a basic background on immune function, the different phases of the immune system, how they react with COVID-19 symptoms and the SARS-CoV-2 virus. There will be an assumed level of knowledge, but I will also try and take it quite slowly. There will be some very basic information in here, so please forgive me if you're a virologist or highly educated medic or clinician who knows everything I'm already talking about. But hopefully we'll find the right balance and we can get some more details in the chat function if we need to later on. So, introducing myself, my name's Martin Lindley. I'm a chartered biologist with the Royal Society of Biology. I'm the Director of Biosciences here at Loughborough at the School of Sport and Exercise Health Sciences. And I'm a member of the Clinical and Translational Research Theme Panel in the Biochemical Society. So that gives me a sort of a broad brush background in biochemistry, the immune function, exercise, etc. Which is why I'm up first really to give this talk. And the idea of this talk really is to give a preparation. James gave you an outline of what's to come as far as nutrition, exercise, well-being, etc. So the idea was to give a basic introduction to the immune system and give you an idea of how it works, and specifically a brief insight into SARS-CoV-2 and the COVID-19 pandemic and how that interacts with the immune system. There are some nuances to this viral outbreak which are very interesting from an immunological perspective and have very important implications for the clinical medical world. So we'll do a little bit about that. So opening up off the top, we have the SARS-CoV-2 virus. This is the virus that is causing all the problems. And I'll make the distinction here that it's the SARS-CoV-2 virus that we're talking about, which is causing the COVID-19 symptoms. So when we're talking virus, this is what we mean. When we're talking symptoms, that's when we talk about the COVID-19. Now, viruses have been open for debate, as it were. One of the biggest debates we have really is whether they are a living entity or not a living entity. Are they alive? What exactly are they? They were first postulated as being poisons, then they were thought of being as being active living organisms. The debate is still out there. I'm not going to get into the debate. All I'm going to say is that they are able to reproduce. They replicate themselves, which means they have RNA material, which means that when they're inside a living host organism, they can replicate and therefore continue their existence, as it were. So the whole point of a virus is it must get into a host, a living host, and that organism must have the mechanisms in place for it to hijack in order for it to replicate itself and continue. Now, one of the reasons that it's been a long while before we got a handle on viruses is that they're extremely small. They're a lot smaller than bacteria. They're a lot smaller than an awful lot of pathogens, and therefore they're very difficult to work with. They're not visible to the human eye, and therefore they are the unseen pathogen, as it were. Now, as I've made a distinction between SARS-CoV-2 and the COVID-19, then we'll start with some terminology. When we first had the news items break about the coronavirus, then it was all over the news that the coronavirus had an outbreak in China. The coronaviruses are actually a large family of viruses. There are lots of different viruses which are all covered under the coronavirus umbrella. The World Health Organization has a few sort of frequently asked questions. These are some of them. The Middle Eastern Respiratory Syndrome, MERS, the Severe Acute Respiratory Syndrome, SARS, are two of the coronaviruses. They're two of the recent ones which have been in the press. 2002, I think, was SARS, the first outbreak. So what's different about this one? Well, they called it a novel coronavirus. So what do they mean by a novel coronavirus? Well, what they meant is they just hadn't seen it before. It's not previously been identified in humans. So this is our first expose, as it were, to this specific coronavirus. Now that has a problem. That means there's an issue. If we haven't been exposed to this virus before on an individual level or on a global level, then we haven't had the opportunity to study it, to actually understand how it works. And if we don't understand how it works, then we have no way of being able to intervene and therefore treat. Now, why have we not seen it before? Why is it novel? Well, it's probably been transferred from an animal base. So we know that the SARS-CoV virus in China in 2002, which was the original SARS outbreak, that seems to have come from civet cats. We know the MERS-CoV virus came from the dromedary camels in Saudi Arabia. Indeed, we know that there is a localized outbreak of MERS on a fairly regular basis because the viral load in the camel population is very, very high. So that it constantly jumps from that population into the human population. Luckily enough, it is fairly localized and we have some ways of managing it. Having said that, there are several known coronaviruses which are circulating in animals which have not jumped. So therefore they have not infected humans. However, we don't know that they won't. What's useful to us is maintaining a constant level of surveillance around the epidemiology of viral infection, specifically coronaviruses, so that we can identify early any potential jumps from animal population to human population. But at the moment, vigilance is about our only protection. So, SARS-CoV-2, the virus that's actually infecting us and giving us our symptoms. As we've said, the virus has an inner material for replication, but the exterior of the virus is probably what's leading it to cause most of our problems. The coronavirus family, and specifically this one, works by entering the host organism and then attaching to the cell surface. And it attaches to the cell surface via a specific protein. We call it a spike protein. And this spike protein will attach to the cell surface via a receptor. So I will just get myself some laser pointers so we're in there. So here's our virus. There's our internal replication material, nucleic acids. Here's our spike protein, which they have many on the surface. And what they do is they attach to specific cell membrane receptors in order for it to begin the process of infiltration into the cell and therefore the replication process. Now, these spike proteins need to be activated, and they're activated via different enzymes or proteins in the cell. The combination of a receptor that's available An activation enzyme, which is in the vicinity, will allow the virus to connect to the cell and then deposit its RNA material into the cell and start the replication process. The SARS-CoV-2 virus, we seem to understand that the ACE2 receptor is the receptor which is allowing it to enter the system. Now, again, this is fairly new information. Most of the virology that we have is post-event. So we find out more about viral infections and pandemics after the event than we do during. We are currently in a race against time to actually work out the mechanisms of action. So a lot of the information I'm giving you at the moment is very not yet confirmed, certainly as the only mechanism, even if we have data to suggest that it is a confirmed mechanism. So we're working in the dark slightly. What we do know is that we have the receptor, and we have a spike protein, and we have an activation enzyme, and we know the SARS-CoV-2 virus is getting into our cells, into our host cell via this mechanism. Now, that leads to the question, can we inhibit this mechanism? And therefore, can we introduce some type of pharmacological agent into the process which will stop the virus from getting into the host and therefore solve all our problems? Unfortunately, it's not that simple. For the clinicians and medics amongst us who understand the ACE receptors, we know that the mechanisms of action, so the angiotensin 2 receptor is a membrane-bound G protein coupled receptor which is involved in the renin-angiotensin system. And therefore, if we interfere with that process or interfere with that system, we'll be having a major impact upon all the metabolic processes and the physiological outcomes from that process. Now, ACE2 receptors are involved in blood pressure, they're involved in blood flow, therefore they're very, very important for some heart conditions and blood pressure conditions. And there are a large number of people who are on medication, whether it be inhibitors or receptors that will actually have an impact upon this. Now, when we found out that the ACE2 receptor was involved, there was a lot of concern whether or not this would have an impact on people who are already on inhibitors or receptor blockers. The current NICE guidelines for the UK and the current World Health Organization scientific note both are very confident to say that there is no, or the evidence does not exist to recommend a change in medication. So for all the clinicians out there who've read about the ACE receptors and are wondering, do we do? The answer at the moment is, do they? There's not enough evidence to make any recommendations for any change in medication. So we stay where we are for now. Now, that doesn't mean that in the future we're not going to have new recommendations. The NICE guidelines are updated regularly. The World Health Organization is adding scientific notes to its websites on a regular basis. So we need to keep vigilant and make sure we keep an eye on this. However, at the moment, the evidence doesn't exist to make a change. So going back to our coronavirus, I mentioned earlier that we have a family of coronaviruses. We mentioned some of them in the MERS and the SARS. The Center for Disease Control in the USA outlines 4 other very common human coronaviruses. Indeed, the NL63, I believe, has a very similar spike attachment protein, which also uses the ACE2 receptor. The OC43 coronavirus, there's a lot of work being done on that because they believe that it is the virus that caused the original Spanish flu outbreak in the early part of the 19th century. So there are other coronaviruses that are circulating. There are other non-coronaviruses that are circulating. What makes the SARS-CoV-2 virus important? One of the reasons is the symptoms it gives and the clinical outcomes. So the symptoms of SARS-CoV-2 are our COVID-19. Now, there's been a lot in the press recently about symptoms. This is the current WHO NHS guidelines, as it were, that clearly outlines that we have a number of different symptoms that are prevalent in our coronavirus: fever, dry cough, tiredness, There are other symptoms that are possible but not common: shortness of breath, aches and pains, sore throat. And there are other symptoms that are common in other viruses, which are not common. They're rare when we have a look at our coronavirus. So the runny nose, nausea, and diarrhea. The diarrhea is something that's actually been picked up recently to say that it might actually be less rare and more common in SARS-CoV-2. But we're waiting for those developments to come online in the next few months. One of the common questions I get asked, in fact, I was asked it by a 9-year-old in the Wellcome Trust, I'm a scientist, get me out of here, is when did the COVID-19 symptoms first appear? Was it in the country before Christmas? And the 9-year-old insisted that it was because he'd had some symptoms. Well, you can see that the symptoms cross all the different viruses. So it could be a common flu virus, it could be a common cold virus, and we're going to have tiredness is going to be across the board. Yeah, aches and pains is going to be across the board. Sore throat is possible and quite common. It's not a very good indicator of the type of viral infection that you have. I know in medicine and clinical terms, we treat the symptoms that are in front of us, but as far as diagnosing what's causing those symptoms, then these broad brush strokes are not very good at identifying which specific virus we're talking about. That being said, self-diagnosis of I've had COVID-19 is probably not a good idea. And if there's any concern at all, then proper antibody testing is the way forward. So that opens us up to an introduction, which has probably been far too long. But now we're going to talk more about the actual immune system itself, what we actually advertised I would talk about. And this is basically how the body is going to fight off the SARS-CoV-2 virus, or indeed any other virus. And what we do is we break down the immune system into two separate processes, which do interact. We only break them down so we can discuss them and talk about them. But we have an innate system, which is a non-specific immediate immune defense. And we have an adaptive system, which is a very specific but much slower immune defense system. So straight into the innate system. As you can see, there's a number of cells within my little red circle. All of these cells are involved in the very complex process of blocking or attacking pathogens that enter the body. These cells are present from birth. They are active straight away. However, they have no memory of any action that they take, and they are not specific to any individual virus, bacteria, or pathogen. However, what they are is very rapid in response and present from birth. So we have a rapid response system, which is a generic block, attack, destroy foreign bodies. However, it has no memory, so it keeps reacting every time it sees something, but it does so very quickly. The second system is an adaptive system. This is a slower process. This is an acquired response, and it's an acquired response to specific individual antigens. These antigens are identified, attacked, and destroyed, and in the process of being destroyed, where the body releases antibodies, which are therefore available for the next antigen response. So there's a memory process. So it's an acquired process. You only get this type of antibody once you've been exposed to the pathogen, but it does have a memory that will respond at a later date much more quickly, but the initial response is very slow. So if we take it into a little bit more detail on the immune system as far as the innate system is concerned, I'm going to break it down into two layers: a physical barrier and a cellular response. It's not a normal way of breaking it down, but I think in the current times, it's probably a good thing to think about. Probably the major physical barrier is going to be the skin, your whole body covering. It stops a virus from entering your cells because it is a physical barrier between the environment and your internal systems. That's a good thing. Unfortunately for us, and I'll put the pointer over your hands, virus will live on for extended periods of time on your hands. It'll live on in any part of the skin, but the hands is a common place for a virus to be found because our nature of our opposable thumbs, therefore we are very dexterous, and therefore our hands go to places that get viruses. Unfortunately for us, we also go through the process of touching our face on a regular basis. So we do this many, many times an hour, and therefore we transfer the virus which hasn't got through the skin, and our barrier has worked, but we then translocate that virus to a mucous membrane, so our eyes or our respiratory tract. If that happens, we still have an opportunity of a physical barrier because we still have our mucous membranes and our ciliated epithelial, which will actually sit there and repel borders, as it were, to a limited degree. Unfortunately for us, the SARS-CoV-2 virus is very small, as all viruses are, and they're very good at connecting to the host cell membrane, and therefore, once they're into the respiratory tract, they're in the respiratory tract. So our best bet is to work with our skin and stop them from entering, which means washing your hands. The more we can do to destroy that virus phospholipid bilayer cell membrane and therefore break it down before it gets in the system, the better. Two ways in which we can do that is heat and soap. Therefore, hot soapy water, washing your hands, is a very effective barrier and destroyer of virus. So, as you can see up here, we have our physical barrier. I said there were two components, the physical and the cellular. Well, the cellular have a huge number of and a huge range of responses and cell types that work very well at controlling and destroying the pathogens that have entered the system. We have phagocytes, cells that remove debris and pathogens. We have an immunological surveillance system, which is our natural killer cells, which will circulate, identify and lyse abnormal cells. We have an interferon system, which is a release of interferons, which will then activate and then supercharge our cells for destruction of the virus. We have a complement system, which attacks and breaks down the cell walls of phagocytes. We have an inflammatory response, which is a very wide-ranging and effective response, and we have fever. Fever is a response. We release pyrogens. The hypothalamus in the brain decides to release pyrogens when we have an infection, and these pyrogens assist in the process of the immune function, but they're also very good as far as being an identifiable symbol of viral infection. So inflammation, the working of the interferons, they're not visible from a clinician standpoint. However, your fever is. So you can take a fever as being a good indicator that there is an infection and therefore an immune response. Unfortunately for us, if you don't have a fever, that does not mean you don't have an infection. If you do have a fever, it is highly likely that you do have an infection. So we mentioned that COVID-19 symptoms from the SARS-CoV-2 virus are very, very specific. We have fever as a common. Our problem at the moment is that we need to work out exactly why the SARS-CoV-2 is so prevalent and so responsive to our bodies, and we have such a wide-ranging pandemic. Now, this is recent research that has been published in the research journal Cell recently, so it's May 2020. The DOI is there. I'll speak to the moderator and get these links put up onto the web page so people can access these references directly. I'll caution this to say that this is one paper and we are still very much working in the dark, but the paper is out there and the evidence does suggest that we have two specific responses from the SARS-CoV-2 virus which are implicating on the innate immune response, which give it its wide-ranging effectiveness as a virus. The first one, SARS-CoV-2 seems to interfere with the process of interferons being released. So in all the different ways in which the innate immune system functions, interferons are released by activated lymphocytes, macrophages, and therefore they then attack the virus-infected cells. This interferon release is vastly reduced by SARS-CoV-2. We don't yet know why. There are some postulated mechanisms, but enough for this talk to say that it reduces the interferons. That's bad because it means the virus is more likely to replicate, and therefore your viral load goes up, and therefore the impact of the virus is going to be magnified. The second response is that the inflammatory response, specifically the cytokine response, which is an inflammatory mediator, that is increased. So it decreases interferons, but it increases cytokines. And this cytokine increase has been termed a cytokine storm, is causing huge problems because the large amount of inflammation beyond which is needed as a response to a virus in normal situations is causing problems in the respiratory system. So our respiratory systems begin to fail because there's a huge amount of inflammation where it shouldn't be, which then leads us into our intensive care situations and assisted ventilation. So 2 ways in which in the innate immune system, the specific SARS-CoV-2 has an impact different from other viruses. Moving on to our adapted or our specific immune function. Again, I've broken it down into cell-mediated and humoral. This is more regular and more normal. Our specific immunity as a cell-mediated revolves around our T cells, which recognize fragments of proteins that are on the antigen and present in a way that we can identify and flag that we have the antigen present or we have an infected cell, and that flag via the MHC, which is our major histocompatibility complex. This basically waves to the rest of the immune system that this cell needs to be destroyed because it's infected. I'm not going to walk you through that diagram. However, I'm going to identify that we have over here our T cell responses, and we have our cytotoxic T cells and our helper cells, which together form the process of destruction of the antigen. So it's our cell-mediated immunity, which is what we're looking at here. I mentioned the MHC as being a flag. So this basically schematic shows that here's our pathogen entering our cell. In this case, it's quite a large pathogen. It enters into the cell and it gets broken down. It gets attacked by the lysosomes. However, fragments of this viral, protein fragments, which can identify which virus it is, get hooked up to MHC proteins, which get transposed to the cell membrane and are therefore external flags which are displayed to the rest of the immune system to identify that this pathogen is around. The humoral antibody-mediated B cells are the second part of the specific immunity. And the B cells can recognize and bind to antigens, specifically in the lymphointerstitial fluid or the plasma itself. And the second half of that same diagram shows our beta cells that start off in the red bone marrow but become mature and are therefore in the circulation. These can identify the antigens, and once the antigen receptors have identified, then we can form proteins called antibodies. These antibodies are they're active and can identify the foreign body, the foreign pathogen, and they're present as a memory cell, so that once you have had the infection and the antibodies have been produced and the virus has been fought off, then the memory cells and the antibodies may continue for a length of time to allow you a level of immunity to the next viral infection. Again, that's just another schematic giving an idea of the process by where our clonal selection and proliferation of B cells start from a single activated cell and they're cloned so that we can have a larger number of active cells, which means we can increase our antibody numbers, which means that we can fight the viral infection. At the same time, we have our memory or our long-lived cells, which will stay in circulation post-viral infection, which can then be primed for the next. This leads us through into a little flow diagram we've got here, where we talk about immunity. When we talk about the immune system, we talk about immunity, and we've talked about the innate immunity, and we talked about the acquired immunity. And if we follow this through, if we have a viral infection that we have at present, individuals become infected, they're exposed, they produce antibodies. These antibodies develop in response to a direct infection, and then we are allowed an active immunity system, so we can have our future referenced, as it were, to the viral impact. We don't seem to have this passive transfer or naturally acquired passive immunity. We're very much following this pathway. Now, as we did for the innate system, we'll do for the acquired. What are the specifics of the immune function response? Well, in this case, it's to do with the antibodies. Again, this is from Nature Medicine. It's a 2020. I think it was last month. Again, I put the caution note up. We are working in the dark. However, the Chinese study seems to suggest that there is an impact of SARS-CoV-2 on antibody production, such that the antibodies that are present for instance, our SARS original outbreak in 2002, they were seen to last for approximately 3 years or so, 34, 36 months. The SARS-CoV-2 antibodies, which have been looked at in very small numbers of patients, so it's again the note of caution, but the antibodies from those infections of people who are recovering seem to start to degrade very quickly and reduced in number within 4 to 6 weeks. That would suggest that we are more likely to have a reinfection rate, we're more likely to have a second wave, and there are concerns about viral load and our ability to fight off a secondary infection. So that's the recent research on the specifics of our SARS as far as the acquired response is concerned. Right, we're already to time, so I'll do a quick wrap up and essentially open the floor for questions. The SARS-CoV-2 virus is a coronavirus. It's a member of one of a large family of coronaviruses. Coronaviruses are not unusual. They are prevalent, and we have large numbers of them that are being studied. SARS-CoV-2 is unusual because it was a novel one. It jumped from an animal system into the human system, and therefore it hadn't presented itself to us before, so we couldn't identify it. We weren't aware of the symptoms and how it related to the infection or how to treat it. COVID-19 symptoms are very severe. That you can go downhill very quickly, and you can go very seriously ill very quickly. The symptoms, however, do range. They are variable. Not all individuals are presenting with exactly the same symptom profile. There are people, and the estimates are a large number of people, who have a viral infection, whose immune system is fighting it off, and therefore they are asymptomatic. The big question is, are the asymptomatic viral carriers able to infect and pass on this, or does the asymptomatic nature of their viral load mean that they are not contagious? We don't know is the answer. In most viral cases, we'd say being asymptomatic doesn't impact. You are able to pass the virus on. There is a window where you can't be contagious, but the evidence simply isn't there for SARS-CoV-2, and I can't give you a recommendation. So, immune system in general, we have our physical barriers, and with this specific virus, the physical barriers are very important, and stopping the virus entering the respiratory system or by the mucous membranes is very important because of the severity of the symptoms. And the physical barrier works well only if we clean that physical barrier on a regular basis. So hot, soapy water breaks down the fatty membrane of the virus and therefore makes it inert. So that is why washing your hands so regularly in hot, soapy water is so important. You are essentially becoming part of your own immune system by providing a physical barrier and destroying the virus itself. Once the virus has entered your mucous membrane, it's gone into the respiratory system, it's in the linings of the lung, then we have an immediate response by the innate immune system. It's not specific, it's a general response. But because it's a general response, it's wide-ranging. There are lots of different cellular structures involved in the process, whether it's a complement system or a phagocyte system, as it were, and they all work together in order to remove that pathogen. At the same time as this innate system is occurring, the acquired system is beginning its process. The innate system works immediately, the acquired takes time. It does the search, it does the identification, but then it doesn't do the flagging because that takes time. So the MHS proteins on the cellular membrane are only out there once they've actually got the virus identified, once they've got the virus killed in the cell structures, and then it's flagged to start identifying it at a later date. So it's a slow process, but it is specific. The benefit of our acquired immunity is that we have our antibodies, which are prevalent in the system for later response. The problem with SARS is that that later response may not be very effective. The evidence suggests that we may not have a very high antibody load post-viral infection. So, checking the time, I think I'm bang on 45. So I will say that the following talks coming up will take you into the realms of nutrition, exercise, specific populations, mental health and well-being, but they're all going to be focused around the impact of a viral load and specifically of SARS-CoV-2. They'll be talking about the symptoms, and they'll be talking about the relevant symptoms to the viral load and the link that we already know exists in general. The ability to talk specifically about this virus is very difficult because it's so new and we're still working on it, but the generic responses in nutrition and exercise seem to hold up. So without further ado, I'd like to thank you all for listening to this talk. I'd like to open up and ask for further questions and to let you know that I'm available via email or on my research group webpage if you wish to discuss further. So with that, I will close down my screen sharing and open it back up for James and our moderators to see what questions we have.
Hi, Martin, can you hear me?
Yep, got you there.
Fantastic. We've got a couple of questions come through on the chat. So okay. The first one is, do we know what success there has been treating COVID-19 with antibodies?
No, we don't, is the quick answer. There are a number of studies going on in the world where we're trying to manufacture antibodies based upon DNA or RNA sequencing of the virus itself. They're all occurring in isolation, as it were, because there's a lot more miss than there is hit in that type of research. So they won't be publishing any type of data until they've actually got some type of positive results. So there are very big, important groups in Oxford, and there's some scattered throughout the world that are working on various different ways of fighting the virus. But as yet, we haven't got that information. So I'm afraid the answer to that one's no.
Thanks, Martin. We've got another question. Does alcohol break down the virus membrane?
Yes, it does. However, it has to be a very, very high proof alcohol, and it doesn't work as well as the soap does. So the alcohol antiseptic gel wipe stuff that people are being pushing through, that does have an effect. It does work. However, it depends upon the level of the alcohol that's present in the gel. The higher, the better. The very high proof alcohol works the best. However, fundamentally, soap actually works better. Hot soapy water is almost a guarantee, as opposed to not quite knowing what proof alcohol you got in the gel. So it's difficult to use hot soapy water all the time, and alcohol rubs do work. It's just that there have been some cases of some very low level alcohol being sold, which isn't going to be effective. So check the proof, the percentage, the higher the better. We want it to be up there in the 90s, but in preference, hot soapy water.
Yeah, fantastic. Another one's just come through. How likely do you think it is that there'll be a vaccine that is successful?
Okay, which piece of string would you like me to tell you the length of next? I mean, if we look at our simple cold virus, then we've known about the cold virus for a very long period of time. It's very prevalent around the world. We're no closer to a vaccine for cold virus than we were when we first identified it. If we look at our flu, we do have vaccines for flu, but we have so many different variants and new strains of flu that governments basically make a decision on which strain of flu that they're going to vaccinate for this year, and it's mathematically modeled based on epidemiological studies, but quite often it gets it wrong and we end up with a very high flu load. There's probably the best chance of us getting a vaccine for SARS-CoV-2 than any other, because the nature of the virus has been so critical. It's been such a devastating pandemic that every lab that can is looking for a vaccine. So the likelihood is high based on other vaccine hunts, but at the same time, other vaccine hunts have gone on for very long periods of time and been unsuccessful. So I can't give you an answer. I can say that there are many, many groups looking for one. It could be 6 months, it could be 6 years, it could be 60 years. I can tell you for sure we will not have a vaccine very soon. Predictions of vaccines being produced and being effective for the summer are vastly overstepping the mark. We may be able to identify candidates, but we won't be able to go full clinical trials and be able to look at effectiveness or indeed the negative impact of side effects. So it's going to be a while before we actually get anything along those lines.
Along the same kind of lines, is the virus still fairly stable, and will this help with the search for a vaccine?
Unfortunately, the reports seem to be that the virus is indeed not stable and it mutates very quickly. So we have identified separate strains of the virus in Italy, in China, in Europe, in America. So it seems to jump quickly, and it seems to be very infectious, and it seems to be very effective at surviving and therefore mutating. And as the question alluded to, this makes it even more difficult to identify a vaccine that will work, because if we're too specific on the vaccine, then it's going to be not very effective across the board. So we have several vaccines for different flu strains. It has taken us a while to get there. We may come up with a vaccine for this particular virus, but it does mutate quite quickly. We do have separate strains identified already. So getting a vaccine or a series of vaccines that work across the board is going to be very difficult and problematic. This really is a unique situation as far as the modern history is concerned. The lockdown occurring at various different places is being eased. That does not mean that the problem is going to go away. This is going to be a viral problem we have for a very long time.
Okay, is COVID-19 more common in certain blood groups?
I don't actually know the answer to that question. It's not something I've come across, so I will write that question down. I will look into it and see what the literature is saying, and then we'll post an answer either on the website and give an indicator at the next talk maybe to see where we can find that answer. It's not something I've seen, but I don't have my finger on the pulse of every area of research. So it's quite likely it's gone by unnoticed from me. So it's something I'll take a look at and see if we can post an answer on that on the website.
Yeah, no problem. We can pick that one up later. Is it possible to have antibodies present in an antibody test without having directly contracted COVID-19?
If the antibody test is specific for SARS-CoV-2, then no, because if you won't have those antibodies, they won't have been produced because your immune system hasn't identified and done the antibody creation. So if you test positive for the antibody, it's highly likely that you have had the viral infection and your body's responded. However, I have a caveat to that. That is in theory. In practice, false positives due to inaccurate tests are very common. Specifically for this virus, because it's new, then antibody tests were rushed to the market. Due process wasn't done very well, and a large number of false positives and false negatives started to occur. So that muddied the water as far as us understanding the nature of the virus and how it progressed. So that again flags and highlights our problem with a successful virus. Without full clinical tests, without rigorous method development and analysis of results, getting things out there quickly is not always the best answer. So in theory, you won't get a positive test because the antibodies won't be there because you haven't had a viral infection. However, the nature of testing means there can be errors in the testing where you get a false positive that says you have the antibodies when you don't actually.
Another question we just had through. How does sweat affect the permeability of skin?
Well, how do I answer this one easily? If your sweat glands are active and your sweat pores are open and you're secreting sweat out into the surface of your skin, then it is giving you an opening for a viral infection. Bacterial infection is quite common in open pore ducts. As far as I know, the transmission of virus into the system isn't likely to be via open sweat ducts. It is much more likely to be via the mucous membranes and the internal lines of the lung. We know it's a lung disease because we have the symptoms present in there. We know it also shifts into a cardiovascular and systemic blood flow, and indeed into the brain and various other tissues. But as far as route of transmission is concerned, I've seen nothing that suggests that it will get into the system via sweat pores. Theoretically, that may be a potential way to get into the system, but in actuality, I haven't seen any evidence to suggest it. So the implication being sweating and doing exercise isn't necessarily a bad thing.
Thank you, Martin. Does strength in personal immunity make it harder to catch?
Well, there's two things there. You've got your initial exposure to the virus. There's lots of things you can do to minimize your exposure to the virus. Washing your hands, social distancing, staying away from areas of known infection, etc., etc. That is the exposure risk. Once the virus is in your system, once the virus has entered your lungs, your respiratory tract, then the strength of your immune system will come into play. If you have a healthy immune system and you can fight instantly and to a very strong response, then you are much less likely to exhibit symptoms and you're much less likely to become ill due to the viral load. So you can minimize the rate at which the virus will actually replicate itself because you're responding quickly and in strength. A weakened immune system will mean that the virus will get into your system and therefore will replicate, viral load will go up, clinical symptoms will increase, and you'll get more ill, more poorly. That is why the shielding process is in place, not just for the elderly, the over 70, who have reduced immune function, but also individuals with specific reduced immune function were isolated and shielded to stop the exposure pattern. Because if you don't get exposed to the virus, then you don't need your immune system to fight it off. You only need your immune system to fight it off exposed to it. And it's always better to avoid exposure than it is to fight the virus internally.
Thanks, Martin. And back to alcohol again. Is a 70% alcohol gel effective?
Well, it will have some effectiveness, but that's not a very high. I'd say go as high as possible. I don't know what the current recommendations are for hospital but I believe they're higher than 70. I think you're looking at the 80s and 90s and getting up as high as possible. It does mean it's more expensive, but it is also more effective. So if you've only got access to 70, that's better than nothing. But that means an awful lot of washing with an awful lot of gel on a regular basis to reduce but not get rid of this risk of exposure.
Thank you. Does chlorine kill the virus? Would swimming be a safe activity to take part in?
Right, well, that's a two-part question. Number one, chlorine does kill the virus. So chlorine kills most things. Part of my research actually looks at inhaled chlorine gas during swimming. It shows that it's not doing your swimmers much good either. But it's used in the swimming pools because it is a very effective disinfectant. That being said, then exercise swimming in a pool would seem to be a very good idea because you're surrounded in chlorine-based water. However, the caveat to that is that in all pools there will be a similar to a meteorological inversion layer, in that there is a water-air interaction, and therefore there's a 6 to 9 inch layer that sits on the top of the pool, which has an awful lot of disinfectant byproducts in it, and also expired gases from people who are swimming in it. So if you're in a swimming pool and you're submerged in chlorinated water, then you're fairly safe as far as the external exposure to that virus is concerned. However, if there is a positive individual who has a viral load, who may be symptomatic or maybe asymptomatic, and they're breathing into that pool air that sits on the surface of the water, then you're going to be exposed to that virus more than if you were out in the great outdoors, breathing the air, as it were, from the outdoors. So I would say that swimming is an excellent form of exercise. Chlorine is an excellent form of getting rid of the virus, but swimming in an enclosed pool environment with lots of people in who may or may not be positive may not be the best idea. If it's an only form of exercise and it's done at a social distance process, I know the pool here at Loughborough has a social distancing process in place where they're minimizing contact and maximizing the benefits of being in the pool, then that would be great. But I'm doing cycling and running and less swimming.
Thanks, Martin. Are we ever going to get a true picture on the prevalence of people who've been infected by the virus? It seems that many who've had the virus confirmed by a PCR test do not produce an antibody response.
Well, I'm not sure that they don't produce the antibody response. I think the recent article I mentioned there from the Chinese group suggesting that the antibody response is severely blunted and therefore is removed in time. So I think they will be responding in some way with an antibody response, but I think the antibody disappears quite quickly by the looks of things. Will we get a good picture? Probably not. The mathematical epidemiological modelers will probably say that they can predict exposure rates quite well, but I think we will end up using the data that we have to mathematically predict, as opposed to doing population-wide testing, which gives us a good picture. It's going to be a long time before we work all this out. As I say, most of the work we have done on global pandemics, it takes us a lengthy period of time before we actually understand how everything happened. We're right in the middle of it at the moment. We are literally fighting fires, and our understanding is growing, and there's a lot of effort being put towards it, but it's going to take a lot of time, and we probably won't get a full picture, but we will have fairly good predictive models.
Fantastic. What factors strengthen or weaken a person's immunity?
Well, I think that's an excellent question, and I think we have a series of talks which are linking up to it very well indeed. There is going to be a talk via Tom Clifford, I believe, who will talk about nutritional strategies that will enhance the immune system. I will take this opportunity to plug one of my areas of research, which is omega-3 polyunsaturated fatty acids, fish oil. A recent review by Professor Phil Calder from Southampton identifies quite clearly from the literature that the omega-3 fatty acids are proactive in enhancing your immune system, your immune function, and our overall immune health. So they moderate the inflammatory response and they mediate those processes. So diet definitely, and Tom Clifford will be giving a talk and we'll be extending that beyond omega-3s into the whole range of different nutritional impacts. Exercise is the obvious one. A level of exercise will enhance your immune system. If you do too little exercise, your immune system will be compromised. If you do too much exercise, your immune system will be compromised. So it is about a dose-response relationship, and it's a moderate volume and a moderate intensity of exercise on a regular basis is what will optimize your immune system and response. High intense, high volume, long durations, that is going to compromise your immune system. That has led to a number of podcasts from the Loughborough experts in sport to talk about specifically elite athletes during this lockdown. The analogy I gave during those podcasts is that you very rarely have a healthy individual at an Olympic Games unless they're spectating. Athletes, people working at the high level, compromise their systems because they're working at the outer limits of their physiological, biological performance. Therefore, their immune system does tend to be compromised. The same for sedentary individuals. If you're not taking any exercise at all, you will be on the low side again. So a moderate activity level with moderate intensity, moderate duration will optimize your immune function.
That's wonderful plugging of your own work there, Martin. Do we know why BAME communities are affected disproportionately?
Well, this is okay. I will temper my response as this is a public lecture, as it were. The data that's been presented has been incompletely analysed. There was a lot of media publication. As far as we can see, there is I believe South Asian population is a highest hit as far as the consequences of viral infection for this virus. However, there was a very large link to comorbidities of diabetes and cardiovascular disease. And it is probably, and I say probably because I can't be definitive, it is probably likely to do with the overall health of the individuals more so than the virus picking specific ethnicities. The outcomes have been worse for the BAM subpopulations, probably because of the overall incidences of diabetes, etc. Again, there is work to show that there is higher incidences of diabetes and cardiovascular disease. Therefore, we should have been aware of this before we looked at our PPEs and looked at our social distancing practices. So this is something that probably could have been foreseen and probably could have been mediated before it happened. But we do seem to have an increased number of BAM population that are suffering disproportionately. The other subpopulation that hasn't been spoken about but was in the same data set that was released was that the males are far more prevalent as far as severe consequences of the virus load. Again, is that because they're male, or is that because of the nature of the cardiovascular disease, the diabetes, and all the other comorbidities? We don't know. So we are working in the dark. Definitive answers are very difficult to come by. We should be doing as much as possible to protect all the individuals in the population. If we can identify specific subpopulations, we should do as much as we can to protect them more so, because the data does seem to show that there are consequences beyond other populations. I can't tell you the mechanisms of actions, but we can tell you that we do already know that there are higher instances of comorbidities, and therefore that gives us a clear indicator that we need to be working in that area.
Is there any evidence that some people have a genetic susceptibility to the virus symptoms?
I would say that I have not seen any evidence. However, I would also say that That's probably because they haven't been doing that work yet, and that we haven't got genome-wide associations for prevalence of symptoms from this viral load. Those studies are not prioritized at the moment. They take a lengthy time to do. So I'd say I haven't seen evidence out there as yet. However, that doesn't mean that the evidence won't be forthcoming. I would say that what we can do is we can identify populations based on a whole host of other things other than genetics, and just work with the information that we have. I say we do know there's a genetic predisposition to comorbidities, diabetes, cardiovascular disease in South Asian populations, so that we can work on that because we know that exists. Does that mean that the virus is linked specifically to the genetics, or whether it's the comorbidity? Then we don't know.
Thanks, Martin. How long can you be infectious for before you show symptoms, and how long may you still be infectious for after you have symptoms?
Again, we're into that piece of string. It's the obvious question. I bet a bottom dollar it's a clinician that's asked, because it's the information we need to know. But what we're seeing with this virus is it's not responding like other viruses. We have a large number of asymptomatic individuals which have viral load. So we know the virus is there, however, they're not symptomatic. The big risk and the big fear was that everybody who had the virus, whether asymptomatic or not, would be carriers and would be able to transfer the virus across the population. That does seem to have occurred. However, it hasn't occurred as drastically as was possible. So I say it's difficult to put that into words. It has been horrendous globally. It's been terrible in this country. However, it could have been a lot worse. So the virus isn't acting as we thought it would, either low transmission or high transmission. So there's still a lot of work to be done to find out who can pass this and transmit this, and at what stage of their symptoms, and at what stage that they clear the symptoms, are they actually safe from transmission. It's, again, we're working in the dark. We're right in the middle of the firefight. We simply do not have that information available. Therefore, acting on the side of caution is the way forward. Reduction of exposure is the way forward. If you have doubts, if there's any concern, then treat as if they are infectious and treat as if they can transmit. Self-isolation, social distancing, all the things that we've been doing, we just need to keep doing them.
What are the associated risks with using an indoor gym to exercise?
Well, essentially, any indoor facility is going to have some form of recirculated air. So if you're in your garage and that's your indoor facility, then you will be rebreathing the air that you breathed out, which is not much of an issue. However, if you're in a public facility and you are in a facility that is recirculating air with alcohol, then you have necessarily increased the risk of the virus entering your respiratory system. This can be moderated by stopping the air circulation. However, this vastly increased temperature and humidity. You can have ventilatory systems where we have a throughput, where they're basically just blowing air unidirectional through the gym. That will reduce it, but you are still sharing a social space. And if you're in the gym, it means you're exercising, which means your ventilation is up. If your ventilation is up, you're more likely to have droplet formation, and therefore you're more likely to be generating aerosols, and therefore you're more likely to be passing the virus. Having said that, it's only over a short distance, hence the 2 meters social distancing and the recommendation for being outdoors. So it really depends on who else is present, what the environment is like, and what you're doing. If you're in the garage by yourself, you're at no greater risk. If you're in a small confined space with lots of other people, you are at much increased risk. If you wish to exercise and you can do so outdoors, that's preferable. If you're going to do it indoors, good ventilation and minimal numbers of people.
Thanks, Martin. Is there any benefit from high doses of zinc and vitamin C in the early stages of infection to assist the body in fighting off the infection?
I will direct the questioner to a fantastic podcast on Expert in Sport with Professor Mike Gleason and myself, where he discussed in detail the zinc supplementation and the vitamin C supplementation. So what we'll do is probably get yourself as moderators to see if we can get those links put up onto the website as they're specifically answering these questions. If you know you have an infection and you know you're in the early stages, then zinc and vitamin C could be useful. However, if you have a varied mixed diet and you have your recommended daily intakes of zinc and vitamin C going into mega doses, may have an effect, but is unlikely. If once the virus has taken hold, then your immune system needs to be operating optimally. Any additional on top of that isn't happening. Having said that, recommendation, it would be to ensure recommended daily intakes. Because we're in a situation where your diet may have changed because you have unavailability of your normal foodstuffs and various supermarkets doing various different switches on you, then what we said at the podcast was a daily supplement, which had multivitamins, where you have your vitamin C, zinc, and various others as a precautionary measure. I know Professor Gleason on the podcast was fairly keen on high levels of zinc, but in most cases you're not going to be able to buy it at the moment because it's sold out, and there's not a huge amount of evidence to say that you're going to be able to identify you have the virus and therefore when you need to take your megadose. So it becomes ineffective if you don't have the right information. So listen to the podcast and daily supplement if you are concerned about your normal diet.
Thanks, Martin. I think this one is the last question. The new guidelines are suggesting 1 metre for social distancing. What is the risk associated with the 1 metre rule?
Right. Well, the 2 metre rule is fairly arbitrary and was designed and put in place from a variety of different perspectives, but mainly on the basis that we needed some rule and we needed some identifier, and we settled on 2 because it gives somebody something to work on. Reducing it to 1 metre, it won't necessarily double the risks, but it will increase the risk slightly. However, 2 meters, 1 meters, 5 meters would be better. 10 meters is even better. So the bigger the distance, the better. Most people, and I can only speak from my experience, but their ability to keep a 2 meter distance is very, very poor. And I think moving it to 1 meter is more of a social, political, allowing opening up of economic status, etc. There will be a slightly increased risk. However, the government, I'm sure, will judge that slightly increased risk to be outweighed by the benefit of society to opening up the economy, etc. If you have any doubt whatsoever and you have any concerns whatsoever, you think you're at risk, you think you're a high population, then I would say then work on your own cognition and stick to a 2 meter.
So I think that's hello. I think that's
James. Yeah, I was just about to say, James, it's over to you.
Yeah, so thank you very much, Martin, for that. I think that talk was absolutely fantastic, and the questions as well were really good. So hopefully that's given everybody a really good introduction to what's to come in the next few weeks. I think Martin mentioned before that he's happy to have any questions. So if you did make a note of his email address, or indeed if you didn't get his email address, we'll put that on the website. So if you do think of any other questions that pop up at any time, hopefully Martin will have some time to write you an email and give you an answer. But we will leave it there for today. Next week, we've got 3 talks related to exercise and immune function, and diet and immune function. So thank you everybody for attending, and yeah, hope to see you all next week.
Thank you very much, James.